Today we are going to talk about a technique called nucleofection. Nucleofection is a technique where we use electroporation to open up the cell membrane and allow for the insertion of a small piece of DNA or RNA, such like siRNA. In order to do this, we need to use our nucleofection machine. So what we're doing here is we're turning on the machine with the switch in the back, wait for the screen to boot up, and then you're going to select the X core unit, which is the unit that we have. Then you're going to select your vessel of choice, and from there you're going to select a compartment and select the program type. Now in our lab we use human keratinocytes, and there happens to be a preloaded program, so scroll to that program and select it to load it into the machine. This will allow you to be ready for when you're ready to insert your cuvettes, ready to go. Next, we're going to review all the equipment that we need. So we need our centrifuge, we need our nucleofection equipment such as the cuvettes and the pipettes, also our solutions which we will combine, our siRNA here in little aliquots of 10 microliters, our media for our cells, and finally our cells themselves. First, what we're going to do is aspirate out the current media in our plates, here shown with using our Pasteur pipettes. And once we aspirate out all the media from our plates, what we're going to do is add trypsin. So there's different types of trypsin, but we use in our lab TripLE, which is a little gentler on the cells than their traditional 0.05% trypsin. So we're going to take our pipette, our serological pipette, and pipette up 3 mils of trypsin per plate, and this is per 10 centimeter plate. We use 3 mils of TripLE to trypsinize our cells. And what we're doing when we trypsinize is we're detaching the cells from the bottom of the plate so we can gather them and do further procedures with them. So when we trypsinize cells, we need to make sure they're back in the incubator at 37 degrees where they're happy. So we place them in the incubator, and after 4 to 5 minutes, we check them under the microscope to ensure that they have been fully trypsinized. So you can see here on our microscope, there's cells that are floating, suggesting that it's been trypsinized. Finally, what we need to do is we need to neutralize the trypsinization process so the trypsin doesn't eat up all our cells using DMEM media. So we add the equivalent amount of DMEM to each plate to neutralize the TripLE, and we're going to wash our plate a couple times with the pipette to ensure that we rinse off all the cells, and then we're going to transfer this supernatant or cell suspension into a 15 mil or 50 mil conical vial, which will allow us to spin down our cells in order to collect a cell pellet. Once we've collected all our cells, we need to spin them down. So what we do is we spin them at 25 degrees at 1500 RPM for 5 minutes. As the cells are spinning, we need to prepare for the next steps of our protocol. First, we're going to add 7 to 8 mils of media, in our case keratinocyte media, which is cell-specific, to new, fresh 10 centimeter plates. We want to do this so that when we do our nucleofection, after we finish our nucleofection, we have somewhere to put our cells, and the cells aren't sitting in the nucleofection buffer for too long, because the nucleofection buffer is actually not healthy for our cells. So right now we're just adding media to our plates and setting these aside in the tissue culture hood. Remember, everything needs to be sterile for next steps. Now, what we also need to do is prepare 500 microliters or half a mil of keratinocyte media to transfer our cells into following nucleofection. So you see here, we're using sterile technique to dump out our microcentrifuge tubes, and as we take out each one, we're going to pour it onto the cap of the container and then close the caps of the microcentrifuge tube immediately so that it stays sterile inside. Next, we're going to label our microcentrifuge tubes, as this is important to keep track of which is control and which is our target gene knockdown, and also which cells we're using, since we have 2 different batches of cells. Next, we're going to take our serological pipettes and add 1/2 ml to each microcentrifuge tube, as demonstrated here. So you get about 500 microliters. So you could give or take like a few microliters. That's not going to affect the process too much, since this is just a recovery step for the cells, not anything that they're going to live in permanently. Once we have done our preparations, our cells should be ready. So here is what keratinocytes look like spun down, and each pellet represents about 1 million keratinocytes. So what we're going to do is we're going to gently aspirate out all the remaining media, careful to not disturb or touch the pellet with your pasteur pipette. Once the media has been aspirated out, we're going to take 100 microliters of our nucleofection buffer, which is a combination of the 2 solutions we saw previously in the video, and we're going to resuspend our pellet in that nucleofection buffer. Make sure when you're working with these samples you're using filter tips, which is good practice when you're working in the tissue culture hood. And so what you see here is we're taking up our 100 microliters of our nucleofection buffer, and we're going to take our cell pellet and resuspend our cells carefully, pipetting up and down about 10 times in order to make sure all the cells have been resuspended. Once you've resuspended your cells in nucleofection buffer, take up your siRNA, which should be in a PCR tube and 10 microliter aliquots, and add your cells to this, ensuring that you pick up all the cell solution from the falcon tube where you spun down your cells. Now, once you add your cells to your siRNA, resuspend at least 10 times using your pipette and gently mixing the mixture, since you want it to be well mixed for the next step. Once this is well mixed, you're going to take your siRNA cell mixture and add it to the nucleofection cuvettes, seen here with the blue cap. What you want to do is add it to the bottom of the tube, ensuring that there are no bubbles at all in your sample. The bubbles will affect the ability to electroporate your cells, so you don't want that to happen, since you want your cells to take up the siRNA as efficiently as possible. Next, we're going to take our cuvette and place it in the vessel, the machine. You press OK, and in 30 seconds or less, you're going to get your cells. And what you do next is you take the pipettes that were set aside at the beginning. You're going to take up that 500 microliters of keratinocyte media we set aside initially when our cells were spinning down, and you're going to add it to the top of this cuvette. And then, using the same pipette, the plastic pipette, you're going to take up the solutions from the bottom. This will ensure that you pick up all the cells in the cuvette and you're not leaving any cells behind. Once you take up this solution, pipette it back into the same Eppendorf tube and place it in the incubator while you work on the remaining samples. Finally, you're going to take your cells that have been recovering in the incubator, you're going to pipette up all the cell solution and add it to the plates that we had prepared previously with the 7 to 8 mils of keratinocyte media. You want to add their cells in a circular motion so they're plated as evenly as possible. Also pipette up and down a couple times with the pipette to ensure that we clean off all cells remaining on the pipette tip. And that is how you do nucleofection. Make sure you put your cells back in the 37-degree incubator so they can grow and be happy, and make sure you check on your cells the next day for any phenotype that could arise. Thank you.